In September 2025, NASA held a press conference about a rock on Mars. Not just any rock, a rock with spots. Dark spots and pale rings scattered across its surface in patterns that the science team called leopard spots. They published a peer-reviewed paper in the journal Nature, and in that paper, they wrote a line that sent shockwaves through the scientific community. They said they found no particularly strong evidence for any non-biological explanation. In plain language, the best explanation for what they were looking at might be ancient life on Mars. At the same time, a telescope a million miles from Earth detected a molecule in the atmosphere of a planet 124 light-years away. A molecule that on our world is only produced by living things. Tonight, we are going to follow these discoveries and ask the question that connects them. What does alien life actually look like? Not science fiction aliens, real ones, based on real science. And the answer is stranger than anything Hollywood has ever imagined. If you enjoy these nighttime documentaries, remember to like and subscribe. It helps us bring you more videos like this. Now, let us begin. Tonight we are going somewhere extraordinary, not backward in time but outward, out past the edge of everything familiar. Out to the places where the question that has haunted our species since the first human looked up at the stars might finally be getting an answer. Are we alone? Over the next few hours, we are going to follow the trail of that question across the solar system and beyond. We will start on Mars, where a robot the size of a car just found something in the mud that made NASA scientists stop and hold their breath. We will travel 124 light-years to a planet twice the size of Earth, where a telescope has detected a molecule that on our world is only made by living things. We will dive beneath miles of ice on a moon of Jupiter, where an ocean larger than all of Earth's oceans combined sits in total darkness, heated from below by volcanic vents on the sea floor. We will visit a moon of Saturn that is actively spraying its ocean into space, handing us samples of its water without us even having to land. We will ask what alien life might actually look like, not the aliens of science fiction, not little green men or tentacled monsters, but what biology, chemistry, and physics tell us life could become on worlds nothing like our own. Worlds where gravity crushes, worlds where methane falls as rain, worlds where the sun never rises because there is no surface to rise over, worlds where plants might be black, where creatures might see in sound, where intelligence might be spread across an entire colony rather than locked inside a single brain. And by the end, you will understand why so many scientists now believe we are on the edge of the most important discovery in human history, or why the silence we find instead might be even more terrifying than any answer. Chapter 1. The Leopard Spots on Mars. In July of 2024, on the floor of an ancient river valley on Mars, a robot named Perseverance stopped in front of a rock. The rock was shaped like an arrowhead. It measured about 3 feet across and 2 feet wide. It was reddish, like most things on Mars. But this rock had something on it that the other rocks did not. Spots, dark spots, tiny ones, scattered across the surface like poppy seeds. And larger spots, pale rings with dark centers, arranged in patterns that the science team would come to call leopard spots. The rock was nicknamed Cheyava Falls. It sat within a geological formation called Bright Angel, a set of rocky outcrops along the edges of Neretva Vallis, an ancient river valley, roughly 400 meters wide, that was carved by water rushing into Jezero Crater billions of years ago. Perseverance had been sent to Jezero specifically because orbital images showed it was once a lake fed by rivers. Where there was water, there might have been life. That was the theory, that was the hope. But for 3 years of driving across the crater floor, Perseverance had found mostly igneous rock, volcanic material. Interesting to geologists, but not exactly what astrobiologists were praying for. Then the rover turned into the river valley and found Bright Angel, and everything changed. The rocks in Bright Angel were different. They were sedimentary, made of clay and silt. On Earth, clay and silt are among the best materials for preserving the remains of ancient microbial life. They trap organic molecules, they lock in chemical signatures. They are, in a sense, nature's filing cabinets, and the Cheyava Falls rock was made of exactly this material. 2 instruments on the rover went to work. The first was called Sherlock, which stands for Scanning Habitable Environments with Raman, and Luminescence for Organics and Chemicals. Sherlock detected organic carbon compounds in the rock, not just traces, the strongest organic detection the entire mission had made. The science team paused. They had a limited number of sample tubes remaining for the entire multi-year mission, and filling one was a serious decision. They decided this rock was worth it. Perseverance drilled a core sample from Cheyava Falls and sealed it in a tube. That sample is now called Sapphire Canyon. It is sitting inside the rover on Mars right now, waiting to be brought back to Earth. The second instrument was called Pixel, the planetary instrument for X-ray lithochemistry. Pixel mapped the precise chemical composition of those strange spots. What it found was striking. The dark poppy seed spots and the larger leopard spots contained minerals called vivianite and greigite, arranged in what scientists call reaction fronts, concentric zones where chemical reactions had played out over time. Now here is why that matters. On Earth, greigite is a mineral that certain microbes produce as part of their metabolism. Vivianite forms in the presence of decaying organic matter. The combination of these 2 minerals, arranged in these specific patterns in a rock made of clay and silt that also contains organic carbon, is a combination that on our planet is primarily associated with biological processes. In September of 2025, more than a year after the sample was collected, the Perseverance science team published a peer-reviewed paper in the journal Nature. The lead author was Joel Hurowitz, an associate professor of geosciences at Stony Brook University. The paper laid out the findings in meticulous detail, and it contained a line that sent shockwaves through the scientific community. The team wrote that they had found no particularly strong evidence for any abiotic explanation for what they were seeing. In plain language, they could not find a good non-biological explanation for the patterns in the rock. NASA held a press conference. The acting administrator called it the closest we have ever come to discovering life on Mars. The project scientist Katie Stack Morgan said that while abiotic explanations cannot be ruled out, they are less likely given the findings. The word used again and again was potential biosignature, a substance or structure that might have a biological origin but requires more study before a conclusion can be reached. Let us be clear about what this is and what it is not. This is not confirmed life on Mars. Nobody is saying microbes definitely lived in Jezero Crater. What the scientists are saying is that they found a rock with a combination of features that on Earth would be considered strong evidence of past microbial activity, and that after a year of analysis and peer review, they cannot find a purely geological explanation that fits as well as a biological one. NASA has a framework for evaluating claims like this. It is called the Confidence of Life Detection Scale, or the CoLD scale. It has 7 benchmarks, a progression from initial detection through increasing levels of confidence. The Cheyava Falls finding is the first result in decades of Mars exploration to even register on it. That alone is extraordinary. But here is the deeper truth that the leopard spots reveal. Even if this particular rock turns out to have a non-biological explanation, even if some clever geochemist figures out a way to produce vivianite and greigite in those patterns without any help from microbes, the finding still changes everything, because it shows us how close we are. We are not talking about life on Mars as a distant hypothetical anymore. We are talking about specific minerals in a specific rock, in a specific ancient riverbed, that might preserve the chemical fingerprints of organisms that lived more than 3 billion years ago. And Mars is not the only place whispering. While Perseverance was drilling into Cheyava Falls, a telescope orbiting 1 million miles from Earth was staring at a planet 124 light-years away. And what it found there might be even more extraordinary. A molecule floating in the atmosphere of an alien world. A molecule that on Earth is produced exclusively by living things. That planet, that molecule, and the firestorm of scientific debate it ignited is where we are going next. Chapter 2. The strongest hint from another world. 124 light-years from Earth, in the constellation Leo, there is a star. It is a red dwarf, smaller and cooler than our Sun. It has a name that sounds more like a license plate than a celestial object, K-18. And orbiting that star, in the zone where temperatures allow liquid water to exist, is a planet that has become the most talked about world in astrobiology. K2-18b was discovered in 2015 by the Kepler Space Telescope. It is big, about 8.5 times the mass of Earth and roughly 2.5 times its diameter. That puts it in an awkward category. Too big to be a rocky world like ours, too small to be a gas giant like Neptune. It sits in a middle ground that scientists have only recently begun to understand. In 2019, the Hubble Space Telescope detected hints of water vapor in the atmosphere of K2-18b. That made headlines. It was the first time water had been found in the atmosphere of a planet orbiting in the habitable zone of its star. But Hubble could only do so much. Its instruments were not powerful enough to see the full picture. For that, scientists needed the James Webb Space Telescope, the most powerful observatory ever built. And when JWST finally turned its golden mirror toward K2-18b, the plot thickened considerably. In 2023, a team led by Niku Madhusudan at the University of Cambridge published the first JWST analysis of K2-18b's atmosphere. They found clear signatures of methane and carbon dioxide. The presence of both, combined with the apparent absence of ammonia, was consistent with a scenario that Madhusudan's team had been developing for years. They proposed that K2-18b might be what they called a Hycean world. The word comes from hydrogen and ocean, a planet with a thick hydrogen-rich atmosphere sitting on top of a global water ocean. An entire world covered in water with no land at all. But the methane and carbon dioxide were not the most exciting part of that 2023 paper. Buried in the data was a weaker signal, a tentative hint of something else, a molecule called dimethyl sulfide, DMS. On Earth, dimethyl sulfide is produced almost exclusively by living organisms. The primary source is marine phytoplankton, tiny photosynthetic organisms floating in the ocean that release DMS as a byproduct of their metabolism. DMS is the molecule responsible for that distinctive smell of the seaside. It plays a role in cloud formation over the oceans, and it has very few known non-biological sources. A hint of DMS on a planet that might have a global ocean was electrifying, but it was just a hint. The signal was weak, not strong enough to be confident. The team needed more data. In April of 2024, JWST stared at K2-18b again, this time for nearly 6 hours as the planet passed in front of its star. The telescope used a different instrument, its mid-infrared spectrograph called MIRI, which looks at a different range of wavelengths than the instruments used in the first observation. When the data came back and the team analyzed it, they found more evidence. In April of 2025, Madhusudan's team published their new results. They reported that the mid-infrared data showed spectral features consistent with dimethyl sulfide and a related molecule called dimethyl disulfide, or DMDS. The detection had reached what scientists call 3 sigma confidence. That means there is roughly a 0.3% probability that the signal occurred by chance. To put that in perspective, The accepted threshold for a confirmed scientific discovery is 5 sigma, which means a 0.00006% probability of chance. 3 sigma is significant. It is enough to make headlines. It is not enough to pop champagne. But the team estimated that with just 16 to 24 more hours of JWST observation time, they might be able to reach 5 sigma. The reaction from the scientific community was immediate and divided. Some praised the power of JWST to even attempt such a detection. Others urged extreme caution. Sarah Seeger, a planetary scientist at MIT, warned that with thousands of exoplanets in view, the temptation to overinterpret is strong. She said some researchers are jumping the gun when it comes to K2-18b, and that enthusiasm is outpacing evidence. And the skeptics had good reasons for caution. In 2024, laboratory experiments demonstrated that DMS can be produced without life under simulated exoplanet conditions. DMS has also been detected in comets and in the interstellar medium, environments far removed from biology. A NASA-led reanalysis published in the summer of 2025 using independent data processing and different atmospheric models found no conclusive evidence of DMS. The debate is ongoing. This is where the story becomes not just about K2-18b, but about something larger. Something that touches the very nature of scientific discovery. Because we have been here before. In 1976, the Viking landers touched down on Mars and performed the first ever experiments designed to detect life on another planet. One experiment mixed Martian soil with nutrients and detected a burst of gas that looked exactly like what living microbes would produce. The principal investigator, Gilbert Levin, went to his grave insisting Viking had found life. Most of the scientific community disagreed. The debate lasted decades. In 1996, a team at NASA announced they had found evidence of fossilized bacteria in a Martian meteorite called ALH 84001. President Clinton held a press conference. The world held its breath. Years of subsequent research cast serious doubt on the biological interpretation. The magnetite crystals that had been the key evidence could have formed without biology. In 2020, a team announced the detection of phosphine in the atmosphere of Venus. Phosphine, like DMS, has few known non-biological sources on rocky planets. The announcement made global headlines. Within months, other teams challenged the detection. Reanalyses using different methods found no significant phosphine signal. The debate continues, but the initial excitement has largely faded. The pattern is consistent. Excitement, caution, debate, ambiguity. Every time we think we have found the smoking gun, the gun turns out to be more complicated than we thought. And that pattern is not a failure of science. It is science working exactly as it should. Extraordinary claims require extraordinary evidence. The bar is high because it has to be. But there is a growing conversation about whether the bar might be too high. In 2025, analysts at RAND published a commentary asking whether the standard for proving alien life is stricter than for other scientific claims. They compared it to the decades it took to establish scientific consensus on human-caused climate change. The question is not whether we should be rigorous. The question is whether our frameworks for evaluating evidence are adequate for a discovery that has no precedent. The philosopher Carol Cleland has framed this as the problem of finding life as we do not know it. Every detection method, every biosignature we search for, is based on the one example of life we have, Earth life. What if alien life uses a different chemistry? What if it leaves no signatures we recognize? What if it is right in front of us and we cannot see it because we do not know what to look for? This is the paradox of the search. We need to find something we have never seen before using tools designed to find only what we already know. K2-18b may or may not have dimethyl sulfide in its atmosphere. It may or may not have an ocean. It may or may not harbor life. But what it has already done is show us that we are living in an era of almost finding life on Mars, almost confirming biosignatures on an exoplanet, almost crossing the threshold from alone to not alone. And that almost is pushing us to ask harder questions, not just about other worlds, but about the one thing we are absolutely certain is alive, the life on our own planet. Because before we can recognize alien biology, we need to understand what makes biology itself tick. What are the rules? What are the exceptions? And most importantly, which features of life on Earth are universal laws of nature and which are just accidents of our particular history? Is less than #0, 5# is greater than? That question, the deepest question in all of biology, is where we turn next. Chapter 3. The one example we have. There is a fact about life on Earth that is so fundamental and so strange that most people never think about it. Every living thing you have ever seen, every tree, every insect, every fish, every bird, every bacterium, every fungus, every human being who has ever lived, shares the same basic operating system. Not similar operating systems, the same one, the same genetic code, the same molecular machinery, the same fundamental chemistry. A bacterium living in a volcanic hot spring and a blue whale cruising through the Pacific use the same 4-letter alphabet to store their genetic information. They build their proteins from the same set of 20 amino acids. They wrap themselves in cell membranes made from the same types of molecules. They run on the same energy currency. 4 billion years of evolution, trillions of species, an explosion of diversity so staggering that no human mind can fully grasp it, and underneath it all, one blueprint, one single experiment in biology. This is the most important thing to understand before we can think seriously about alien life. We have exactly one example of life in the entire universe, a sample size of 1, and everything in that sample traces back to a single common ancestor, some simple cell that lived roughly 4 billion years ago in conditions we can only guess at. Every living thing on Earth is a descendant of that cell. Every instance of biological innovation we observe happened within the same family tree. That means we cannot know which features of Earth life are necessary for any life and which are just the way things happen to work out here. Is carbon essential? Or did life on Earth use carbon because carbon was abundant in the environment where it started? Is water essential? Or is it just the solvent that happened to be available? Is DNA the only way to store genetic information? Or is it one solution among many? Let us look at what Earth life actually runs on. The first pillar is carbon chemistry. Carbon is the supreme molecule builder of the periodic table. It can form 4 bonds simultaneously. It can link with itself to create chains, rings, sheets, and 3-dimensional frameworks of essentially unlimited complexity. No other element comes close to this versatility. The number of known carbon compounds exceeds 10 million. The number of possible carbon compounds is effectively infinite. This is why carbon sits at the center of every biological molecule on Earth. Proteins, fats, sugars, DNA, RNA, all of them are carbon-based. The framework that holds them together, the skeleton that gives them shape, is built from chains of carbon atoms bonded to each other and to hydrogen, oxygen, nitrogen, sulfur, and phosphorus. The second pillar is water. Water is strange. It is one of the few substances that expands when it freezes, which is why ice floats. If ice sank, lakes and oceans would freeze from the bottom up, killing everything in them. Water has an unusually high heat capacity, meaning it absorbs and releases heat slowly. This stabilizes temperatures, preventing wild swings that would destroy delicate chemistry. Water is an excellent solvent, dissolving more substances than almost any other liquid. This allows the complex chemical reactions of life to take place in solution, with molecules meeting and interacting freely. Every biochemical reaction in your body happens in water. Every cell is filled with it. Every organism on Earth depends on it. Life as we know it is inseparable from water. The third pillar is the genetic code, DNA and RNA, two long chain molecules that store and transmit information using a four-letter chemical alphabet: adenine, thymine, guanine, cytosine. That is it. Four chemical bases arranged in sequences billions of letters long. Those sequences encode the instructions for building every protein, every enzyme, every structural component that a cell needs to function. The code is universal, not just similar across species, identical. The same three-letter combinations that code for the amino acid leucine in a bacterium code for leucine in a human. The same molecular machinery that reads the code and translates it into proteins works the same way in every organism ever studied. This universality is the strongest evidence we have that all life on Earth descends from a single origin. The fourth pillar is cellular organization. Life does not just happen in open solution, it happens inside cells. Tiny compartments, bounded by membranes that separate the interior chemistry from the outside environment. The membrane is not passive. It actively controls what goes in and what comes out. It maintains concentrations. It creates gradients. It is a sophisticated gatekeeping system that allows the cell to maintain its internal order against the constant pressure of entropy. These four pillars, carbon chemistry, water, genetic code, cellular organization, together they define life as we know it. And every organism on Earth, from the simplest archaea to the most complex animal, runs on all four. Now here is the uncomfortable question. Are these four pillars the only way to build life? Or are they just the way our life works? We genuinely do not know. And the reason we do not know is that sample size problem. We have one tree of life, one data point. You cannot establish a universal law from a single example. You can observe what is. You cannot determine what must be. Some scientists have even asked whether a second origin of life might have occurred right here on Earth. The shadow biosphere hypothesis proposes that alternative life forms using different chemistry could exist alongside us undetected because we have never looked for them. Every technique we use to detect and study life assumes familiar biochemistry. If something were alive but built differently, our instruments might not recognize it. Our tests might miss it. We might have been walking past it our entire history as a species. No evidence for a shadow biosphere has ever been found. But the hypothesis highlights a critical point. Our understanding of life is based entirely on one version of it. We are prisoners of our own biology. NASA, to its credit, has tried to define life in a way that does not assume Earth chemistry. Their working definition is elegant and deliberately broad. Life is a self-sustaining chemical reaction capable of Darwinian evolution. Notice what that definition does not say. It does not say carbon. It does not say water. It does not say DNA. It describes a process, not a substance. Any system that can sustain itself chemically and evolve through variation and selection qualifies. By this definition, life built on entirely different chemistry, using different solvents, storing information in different ways, would still be life. In 2024, a gathering of scientists explored an even more radical idea. What if evolution itself is not limited to biology? What if it is a universal law that applies to any sufficiently complex system? Chemical systems, mineral systems, information systems. The concept, sometimes called assembly theory, proposes that the complexity of a molecule or structure can itself serve as a biosignature. The idea is that beyond a certain threshold of complexity, random chemistry cannot produce the observed structures. Only a system undergoing selection, some form of evolution, can build things that complex. If assembly theory holds, it would give us a way to detect life without knowing its chemistry. We would not need to look for specific molecules. We would look for complexity itself. We would look for order that cannot be explained by physics and chemistry alone. This is still theoretical. Assembly theory is debated, but it represents the cutting edge of how scientists are thinking about the search for life. Not searching for what we know, but searching for patterns that transcend any particular chemistry. And we are going to need that kind of thinking. Because when you look at what life has managed to do on our own planet, when you see where it has managed to survive and thrive, you realize that the limits of biology are far wider than anyone imagined even a few decades ago. In 1977, a submersible descended to the bottom of the Pacific Ocean and found something that rewrote the rules of life on Earth. It found organisms thriving in total darkness, at crushing pressure, in water hot enough to melt lead. Organisms that had nothing to do with the sun. Organisms that drew their energy from the Earth itself. That discovery and the cascade of revelations that followed it is where we go next. Chapter 4. Life in the Dark. For most of human history, we believed one thing about life with absolute certainty. All life depends on the sun. Plants capture sunlight. Animals eat plants, or eat other animals that eat plants. The entire web of life is powered from the top down by solar energy. No sun, no life. That was the rule. In 1977, the rule was broken. A team of scientists aboard the research submersible Alvin descended to the Galapagos Rift, a volcanic spreading center on the floor of the Pacific Ocean, roughly 2.5 kilometers below the surface. They expected to find a barren landscape. The deep ocean floor, far from sunlight, was supposed to be a biological desert. A few scattered organisms scraping by on the thin rain of dead organic matter drifting down from the sunlit waters above. That was the prediction. What they found instead was one of the most astonishing scenes in the history of biology. Around cracks in the ocean floor, superheated water was gushing from volcanic vents. The water was black with dissolved minerals, giving these formations their name, black smokers. The temperatures at the vent mouths exceeded 300 degrees Celsius. The water was loaded with hydrogen sulfide, a gas that is toxic to most life on Earth. The pressure was immense. There was no light. None. Total, absolute darkness. And surrounding these vents, packed against them, living in them, was an ecosystem of staggering abundance. Giant tube worms up to 2 meters long with bright red plumes, clams the size of dinner plates, dense swarms of shrimp, thick mats of bacteria coating every surface, an entire food web thriving in conditions that should have been lethal. The key was the bacteria. These microbes were not photosynthetic. They did not need the sun. They drew their energy from a completely different source. They oxidized hydrogen sulfide pouring from the vents, using the chemical energy released by that reaction to build organic molecules from carbon dioxide. The process is called chemosynthesis. It is the mirror image of photosynthesis, except the energy source is not light. It is chemistry, geology, the heat and minerals of the Earth itself. The tube worms had no mouth, no stomach, no digestive system of any kind. They did not eat. Instead, they harbored billions of chemosynthetic bacteria inside their bodies in a specialized organ called the trophosome. The bacteria fed the worms. The worms provided the bacteria with a stable environment and a steady supply of chemicals. It was a partnership, a symbiosis so complete that neither organism could survive without the other. The discovery of hydrothermal vent ecosystems was a revolution. It proved that life does not need the sun. It does not need photosynthesis. It does not need the surface. All it needs is liquid water, a source of chemical energy, and the right raw materials. And that changes everything about the search for life elsewhere, because the sun is just one star among hundreds of billions. But geology, water, and chemistry are everywhere. Since 1977, the story has only gotten stranger, because the hydrothermal vents were just the beginning. The real surprise was what scientists found when they started looking deeper, not deeper in the ocean, deeper in the rock, kilometers below the surface of the Earth, in solid rock, in environments with no connection to the surface, no sunlight, no oxygen, no organic matter drifting down from above, scientists found life. They are called the deep biosphere, microbial communities living inside the crust of the planet itself. Some of these organisms exist at depths of 5 kilometers or more. They live in the microscopic spaces between mineral grains, in fractures filled with ancient water, in environments that have been sealed off from the surface for millions of years. How do they survive? The answer is remarkable. Radioactive elements in the surrounding rock, uranium, thorium, potassium, undergo natural decay. The radiation from this decay splits water molecules into hydrogen gas and oxygen. The microbes use the hydrogen as fuel. They combine it with carbon dioxide or with dissolved minerals to generate the energy they need to survive. They are powered by radioactive decay. They are eating radiation, in a sense, and they could theoretically survive indefinitely. As long as the rock contains radioactive elements, and virtually all rock does, these organisms have a power source. They do not need the sun. They do not need the surface. They do not need anything except geology and water. In 2025, astrobiologists at the University of Colorado Boulder published new research on a process called serpentinization. This is what happens when iron-rich minerals deep in the Earth's crust react with water. The reaction produces hydrogen gas and heat, and it creates vast microbial biospheres thriving in the rock, communities of organisms living entirely on the chemical energy produced by the interaction of water and stone. The implications for the search for alien life are profound. Any rocky planet or moon, anywhere in the galaxy, could potentially harbor life deep in its crust. It does not need to be in the habitable zone of its star. It does not need liquid water on its surface. It just needs liquid water somewhere underground, and rock that can react with it. Even rogue planets, worlds that have been ejected from their solar systems and drift through interstellar space with no star at all, could potentially have living interiors, heated by radioactive decay, insulated by thick crusts of ice and rock, with liquid water trapped deep inside, worlds of eternal darkness, billions of miles from the nearest sun, and potentially alive. The deep biosphere is not a curiosity. Scientists estimate that the total mass of microorganisms living underground may rival or exceed the total mass of all life on the surface. We live on top of a planet that is, quite literally, alive all the way down. And then there are the extremophiles, the organisms that have proven, over and over again, that every assumption we make about the limits of life is wrong. Deinococcus radiodurans, a bacterium that can survive radiation doses thousands of times the lethal level for a human. It has been found living inside nuclear reactors. Its DNA repair mechanisms are so efficient that it can reassemble its own shattered genome after being blasted with enough radiation to sterilize a hospital. Tardigrades, microscopic animals, sometimes called water bears, that can survive the vacuum of space. They have been exposed to temperatures near absolute zero and temperatures above 150 degrees Celsius. They survive pressures 6 times greater than the deepest ocean trench. They survive radiation. They survive desiccation, drying out completely, and then rehydrating years later and walking away. In 2019, an Israeli spacecraft crashed on the moon with tardigrades aboard. Scientists believe some of them may still be viable. Thermophiles living in hot springs in Yellowstone National Park at temperatures exceeding 100 degrees Celsius. Psychrophiles surviving in Antarctic ice at temperatures far below freezing. Acidophiles thriving in water as acidic as battery acid. Barophiles in ocean trenches under pressures that would crush a submarine. Every time we draw a line and say, life cannot survive beyond this point, some organism crosses it. Every boundary we set gets pushed back. The lesson is consistent, and it is humbling. Life finds a way. Not just on the surface, not just in the oceans, everywhere. In 2025, James Holden, a researcher at the University of Massachusetts Amherst, published work on building laboratory environments that mimic the conditions inside Europa, the ice-covered moon of Jupiter. He used deep-sea vent microbes from Earth as test subjects. His conclusion was that if life exists on Europa, it should look something like our own hydrothermal microbes. The chemistry works, the energy is there, the conditions match. And Europa is not the only hidden ocean in our solar system. In fact, the more we look, the more we find them. Oceans of liquid water buried beneath shells of ice on moons that were once considered dead frozen rocks. One of those moons has a spacecraft heading toward it right now, the largest interplanetary probe ever built, carrying 9 science instruments and a radar system that just proved it can see through ice. That mission, those oceans, and what they might contain, is what awaits us in the next chapter. Chapter 5. The Hidden Oceans. If you asked most people where to look for alien life in our solar system, they would say Mars. And Mars is a reasonable guess. It had water, it had a thick atmosphere, it might have had life. But Mars is dry now. Whatever water once flowed on its surface has been gone for billions of years. If life existed there, it is almost certainly dead. Fossils at best. Chemical ghosts in the rock. The most exciting places to look for living alien organisms right now are not planets at all. They are moons. And the most exciting of those moons orbits Jupiter. Europa is slightly smaller than our own moon. Its surface is a shell of ice, smooth and bright, crisscrossed by a web of reddish-brown fractures that give it the appearance of a cracked egg. The surface temperature hovers around -160 degrees Celsius. It is, at first glance, one of the most inhospitable places in the solar system. But beneath that ice is an ocean. We have known about Europa's ocean since the late 1990s, when NASA's Galileo spacecraft detected something unexpected during repeated flybys. Europa has an induced magnetic field. That magnetic field changes in response to Jupiter's powerful magnetosphere in a way that can only be explained by a global layer of electrically conductive fluid beneath the surface. Salt water. Subsequent observations supported the finding. The Hubble Space Telescope detected what appeared to be plumes of water vapor erupting from Europa's surface, though these observations remain debated. The pattern of fractures on the surface is consistent with a shell of ice floating on liquid and being constantly cracked and refrozen by tidal forces. The ice shows signs of being geologically young in many places, meaning the surface is being constantly renewed. Something beneath is pushing fresh material to the top. The numbers are staggering. Europa's ocean is estimated to be 50 to 150 kilometers deep. It contains 2 to 3 times more water than all of Earth's oceans combined. And it is not just water sitting passively in a frozen shell. Jupiter's immense gravity flexes Europa like a rubber ball as the moon orbits, and that flexing generates heat, tidal heating, the same process that drives the volcanoes on Jupiter's moon Io. On Europa, that tidal heating is believed to drive volcanic activity on the ocean floor. Hydrothermal vents, exactly the kind of environment where life thrives on Earth. Hot water, dissolved minerals, chemical energy, all in a global ocean shielded from radiation by kilometers of ice. In January of 2026, scientists published a finding that made Europa's ocean even more interesting. They demonstrated that salty, nutrient-rich surface ice on Europa can become dense enough to break free from the shell and sink downward through the ice. The ocean is not isolated. It may be getting fed with materials from the surface, chemicals that have been processed by Jupiter's intense radiation environment. If that is true, it means Europa's ocean could have a richer chemistry than a sealed system would produce on its own. And we have a spacecraft on its way to find out. Europa Clipper, launched on October 14, 2024, on a SpaceX Falcon Heavy rocket. It is the largest interplanetary probe NASA has ever built. Its solar arrays span 30 meters. It carries 9 science instruments designed to study Europa in more detail than any previous mission could. In March of 2025, Europa Clipper performed a gravity assist flyby of Mars using the planet's gravitational pull to reshape its trajectory toward Jupiter. During that flyby, the mission team conducted a critical test. They activated the spacecraft's radar instrument, called REASON. That stands for Radar for Europa Assessment and Sounding, Ocean to Near Surface. REASON is designed to see through Europa's ice shell. It sends radio waves down through the ice and listens for echoes bouncing back from subsurface structures, pockets of water inside the ice. The boundary between the ice shell and the ocean below, features that could tell us how thick the ice is and where the ocean might be closest to the surface. Testing this radar required a real planetary surface to bounce signals off of, and there is no way to build a test chamber large enough on Earth. Mars provided the perfect target. The radar was activated for about 40 minutes as the spacecraft descended from roughly 5,000 kilometers to 884 kilometers above the Martian surface. It collected 60 gigabytes of data and produced a detailed radargram of Mars terrain. The principal investigator, Don Blankenship of the University of Texas at Austin, said they got everything out of the flyby that they dreamed. Every part of the instrument proved itself. Europa Clipper will perform one more gravity assist, swinging past Earth in December of 2026, before heading to the outer solar system. It is expected to arrive at Jupiter in April of 2030. Once there, it will not orbit Europa directly. Instead, it will orbit Jupiter and make 49 close flybys of Europa, some passing within just 25 kilometers of the surface. Each flyby will activate all 9 instruments simultaneously, building a comprehensive picture of the Moon over time. And here is the tantalizing possibility. If Europa's plumes are active when Clipper arrives, the spacecraft could fly directly through erupting water vapor and sample Europa's ocean without ever landing. A taste of an alien sea from orbit. But Europa is not the only ocean moon in our solar system, and arguably, it is not even the most promising one. 500 kilometers across, that is how small Enceladus is. A moon of Saturn so tiny you could drive across it in a few hours if there were roads. It is smaller than the state of Arizona, and it may be the most important world in the solar system for the search for life. In 2005, the Cassini spacecraft flew past Enceladus for the first time and discovered something astonishing. At the moon's south pole, from a set of parallel fractures in the ice that scientists named tiger stripes, plumes of water ice were erupting into space. Enceladus was geologically active. It was venting its interior into the vacuum. Over the next 12 years, Cassini flew through those plumes multiple times, sampling the material directly. What it found reads like a checklist for habitability: water, organic molecules, salts, silica particles that on Earth only form in hydrothermal systems at specific temperatures. Molecular hydrogen, which, on our planet, is produced by hydrothermal vents and consumed by microbes as fuel, carbon dioxide, ammonia, and then in 2023, using archived Cassini data that took years to fully process, scientists announced the detection of phosphorus, sodium phosphates locked in ice grains from the plume. Phosphorus is the rarest of the 6 essential elements for life: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Scientists refer to them collectively as CHNOPS. With the phosphorus discovery, Enceladus became the only body in the solar system besides Earth with confirmed evidence of all 6 CHNOPS elements in a liquid water environment. The concentration of phosphorus was estimated to be at least 100 times higher than in Earth's oceans. The implications are extraordinary. Enceladus has liquid water, it has hydrothermal activity, it has all the chemical building blocks for life, it has a source of chemical energy. and it is handing us samples of its ocean for free, spraying them into space where a spacecraft can simply fly through and collect them. The European Space Agency is now planning a dedicated mission to Enceladus. A spacecraft would not need to land. It would not need to drill through ice. It would just need to fly through the plumes with the right instruments. Future mass spectrometers, far more sensitive than what Cassini carried, could potentially identify not just the ingredients for life, but the products of life. Amino acids in specific arrangements. Lipids with biological signatures, perhaps even fragments of cells. We are standing at the threshold of a search that our grandparents could only dream about. Robots on Mars examining rocks that might preserve ancient microbes. A telescope a million miles from Earth detecting molecules on distant planets that only living things produce here. A spacecraft hurtling toward an ocean hidden beneath miles of ice on a moon of Jupiter. A tiny moon of Saturn spraying its ocean into space as if daring us to come and look. But what would alien life actually be made of? Not all life needs to run on the same chemistry as ours. Some of the most mind-bending science of the last decade has been exploring alternatives, other elements, other solvents, other worlds where the rules are completely different. That is where we go next, into the chemistry of the truly alien. Chapter 6. The chemistry of the alien. Everything alive on Earth is built from carbon, but carbon is not the only element that can form the kind of complex molecular structures that life requires, and water is not the only liquid that can host chemistry. If we are serious about imagining what alien life might look like, we need to break free from the assumption that biology must work the way it works here. The most frequently discussed alternative to carbon is silicon, and the comparison is not arbitrary. Silicon sits directly below carbon on the periodic table. Like carbon, it forms 4 bonds. Like carbon, it can link with itself to form chains. Like carbon, it bonds readily with oxygen and hydrogen. In the broadest chemical sense, silicon can do many of the things carbon does. But the differences are significant. Silicon to silicon bonds are weaker than carbon to carbon bonds. They break more easily. Long silicon chains are less stable than long carbon chains, which limits the complexity of the molecules silicon can build. And there is a bigger problem. When carbon reacts with oxygen, it produces carbon dioxide, a gas. When silicon reacts with oxygen, it produces silicon dioxide, sand, a solid. On Earth, carbon-based organisms breathe out carbon dioxide as a waste product, and it drifts away. A silicon-based organism would exhale sand. Its waste product would not disperse, it would accumulate. This creates fundamental metabolic challenges that have led many scientists to dismiss silicon life as implausible. But not everyone agrees. In 2016, researchers at the California Institute of Technology achieved something remarkable. Francis Arnold, who would later win the Nobel Prize in Chemistry, led a team that engineered bacteria to create silicon-carbon bonds, bonds that do not occur in nature on Earth. The bacteria, modified through directed evolution, incorporated silicon into organic molecules, proving that the barrier between carbon chemistry and silicon chemistry is not absolute. Life, even Earth life, can be coaxed into working with silicon if given the right push. And in 2023, a study at the University of Wisconsin-Madison discovered something even more provocative. Researchers led by Betul Kakar found 270 different autocatalytic reaction cycles. These are self-sustaining chemical reactions, reactions that feed on their own products to keep going. Many of these cycles involved non-carbon chemistry. The findings suggested that self-sustaining chemistry, which is a fundamental requirement for life, may not be as rare or as carbon-dependent as we assumed. The most realistic scenario for silicon biology may not be pure silicon life, but hybrid organisms, life that uses both carbon and silicon in its molecular architecture. Creatures with mineral-like exteriors, and organic interiors, crystalline structures integrated into biological systems. On a world with abundant silicon and the right conditions, evolution might find ways to incorporate silicon that we have not imagined. But alternative biochemistry is not just about swapping one element for another. It is also about the liquid that hosts the chemistry. On Earth, that liquid is water. But water is not the only solvent in the universe. Consider ammonia. Like water, ammonia is a polar molecule. It can dissolve many substances. It remains liquid at much lower temperatures than water, between -78 and -33 degrees Celsius at standard pressure. On a cold world where water would be frozen solid, ammonia could serve as a liquid medium for chemistry. Proteins might fold differently in ammonia. Cell membranes would need different structures. But the basic processes of life, dissolving, reacting, building, could potentially proceed. Then there is methane. Liquid methane exists on the surface of a real world in our solar system right now. Titan, the largest moon of Saturn, has lakes and seas of liquid methane and ethane. It has a complete weather cycle, with methane clouds, methane rain, and methane rivers carving channels across its surface. The surface temperature is -179 degrees Celsius. Water is frozen as hard as granite. In 2015, researchers at Cornell University proposed a theoretical structure they called an azotosome, a cell membrane that could function in liquid methane. On Earth, cell membranes are made of phospholipids, molecules with a water-loving head and a water-repelling tail. They spontaneously organize in water to form the barriers that define cells. An azotosome would work on the same principle but using nitrogen-based molecules instead of phospholipids, organized to function in liquid methane rather than water. The chemistry is radically different. Methane is nonpolar, so the molecular rules for building structures in it are essentially reversed from water. A cell in liquid methane would be built inside out by Earth standards, but the mathematical modeling showed it could work. Stable, flexible membranes could form. The building blocks exist in Titan's atmosphere. Then there is sulfuric acid. The clouds of Venus contain droplets of concentrated sulfuric acid. For decades, scientists wondered whether microbial life could survive in those clouds. The 2020 phosphine detection, though still debated, briefly made Venus the most talked about target in astrobiology. Sulfuric acid is a powerful solvent. It remains liquid over a wide temperature range. On Venus, the cloud layer between roughly 50 and 60 kilometers altitude has temperatures and pressures remarkably similar to Earth's surface. If organisms could tolerate the acidity, the environment is not as hostile as it sounds. And there are even more exotic possibilities. Supercritical fluids. When carbon dioxide or hydrogen is heated and pressurized beyond a critical point, the distinction between liquid and gas disappears. The substance becomes a supercritical fluid with properties of both. It can dissolve things like a liquid and diffuse through materials like a gas. Some researchers have speculated that supercritical fluids on high-pressure worlds could serve as solvents for forms of chemistry we have never encountered. Each alternative solvent would produce life built in ways we have never imagined. Organisms in liquid methane would operate at temperatures where Earth chemistry barely functions. Their metabolism would be incredibly slow by our standards. A single heartbeat, if they had hearts, might take hours. A generation might last centuries. But on their timescale, they would be perfectly normal. And we have a chance to look for exactly this kind of life. NASA's Dragonfly mission is a rotorcraft drone designed to fly across Titan's surface. It will launch in the late 2020s and arrive at Titan in the mid-2030s. Titan's thick atmosphere, about 1.5 times the surface pressure of Earth, and its low gravity, roughly 1/7 of Earth's, make flight easier there than on our own planet. Dragonfly will hop from site to site, analyzing surface materials, measuring chemistry, and searching for the building blocks of life in an environment completely unlike anything on Earth. If life exists in Titan's hydrocarbon lakes, It would be the most profound discovery in the history of biology. Not just because it would prove we are not alone, but because it would prove that life is not locked into one chemical pathway. It would mean biology can work in ways fundamentally different from everything we know. Carbon and water would be revealed as one solution among many, and the universe, already impossibly vast, would become even more impossibly full of possibility. The philosophical stakes are enormous. Finding microbial life on Mars or Europa that uses familiar chemistry would be revolutionary, but it would leave open the question of whether all life must be built the same way. Finding life on Titan, built on completely different chemistry, would close that question permanently. It would prove that the universe generates life through multiple pathways, that biology is not an accident. It is inevitable. But chemistry is not the only force that shapes what life can become. There is another force, so fundamental and so inescapable that we barely think about it. A force that dictates whether a creature walks or crawls, whether it flies or sinks, whether it grows tall or stays low. A force that would sculpt alien bodies in ways we can calculate but can barely imagine. That force is gravity, and it shapes everything. Chapter 7. Gravity shapes everything. You do not notice gravity because you have never lived without it. It is the most constant force in your life. It pulled you into the world the day you were born, and it will hold you down until the day you die. Every bone in your body, every muscle, every blood vessel, every aspect of your anatomy exists in its current form because evolution shaped it under one specific gravitational pull, Earth's 9.8 meters per second squared. Change that number and everything changes. We now know that the galaxy is full of planets, and those planets come in a staggering range of sizes. Super-Earths, rocky worlds larger and more massive than our own, are among the most common types of planet detected by our telescopes. Thousands have been found. Many sit in the habitable zones of their stars. If life exists on any of them, it evolved under gravitational forces that would crush a human being. Consider a world with twice Earth's surface gravity. Not a gas giant, a rocky world with a solid surface, just more massive. On this planet, everything weighs twice what it weighs here. Every step requires twice the effort. Every heartbeat must pump blood against twice the resistance. The bodies that evolve under these conditions would be shaped by that relentless downward pull. Shorter, stockier, thicker bones, denser muscles, more powerful cardiovascular systems. Tall, thin body plans would be structurally unsound. Bipedalism, walking upright on two legs, might never evolve because the engineering becomes precarious. A two-legged creature at twice Earth's gravity needs enormously thick legs to support its weight, and a fall would be devastating. 4 legs, 6 legs, or body plans that keep the center of mass low to the ground become far more likely. Slithering, like a snake, becomes an extremely efficient mode of movement in high gravity. No legs to support, no risk of falling. The body distributes its weight across a large surface area. On a super-Earth, the dominant large predators might be serpentine rather than bipedal. Flight becomes much harder. The energy required to stay airborne scales with gravity. On a world with twice Earth's pull, a flying creature would need proportionally larger wings or a more powerful metabolism, or both. Flight is not impossible, but the size limit for flying animals would be much lower than on Earth. No giant birds, no pterosaurs. Flying creatures on a high-gravity world would tend to be small and powerful, built like bullets with wings. Unless the atmosphere is thicker, a denser atmosphere provides more lift, which partially offsets the increased gravity. A super-Earth with a thick atmosphere could still produce large flying organisms, but they would be shaped differently. Broader wings, more buoyant bodies, designs that exploit aerodynamic lift in dense air rather than fighting gravity with raw muscle power. The landscape itself would be different. Erosion acts faster under higher gravity. Rivers cut deeper. Mountains are ground down more quickly. The terrain would tend to be flatter, smoother, with shallower topographic variation. A high-gravity world might look like an endless rolling plain, with wide, deep river valleys but few towering peaks. Now flip the equation. Imagine a world with low gravity. Mars has 38% of Earth's gravity. Europa has just 13%. The Moon has 16%. On a low-gravity world, the constraints that limit size on Earth relax dramatically. The square-cube law, the principle that says a creature's weight increases faster than its structural strength as it grows, becomes far less punishing. Trees could grow hundreds of meters tall with slender, graceful trunks. Animals could have long, delicate limbs, thin bodies, elegant proportions that would be impossible under Earth's pull. Flight becomes trivially easy, smaller wings relative to body size, less energy required. On a world with 1/3 Earth's gravity and a thick atmosphere, organisms the size of whales might fly. The sky could be as rich with life as the ocean is on Earth. But low-gravity creatures would be fragile by our standards. Their bones would be thinner, their muscles weaker. A low-gravity organism visiting Earth would be crushed by its own weight, unable to stand, unable to breathe against the resistance of its own chest. We are adapted to our gravity. They would be adapted to theirs. Neither could survive in the other's world without technological assistance. And then there are the truly alien environments. Worlds where gravity is not the only variable. Worlds where there is no solid surface at all. In 1976, Carl Sagan and astrophysicist Edwin Salpeter published a speculative but scientifically grounded paper about life in the atmosphere of Jupiter. Not on Jupiter's surface, because Jupiter may not have a surface in any meaningful sense. It is a gas giant, a world of immense atmospheric layers where pressure and temperature increase as you descend. Somewhere deep inside, the atmosphere transitions into a supercritical fluid and then into liquid metallic hydrogen. There is no ground to stand on. But there are layers, zones where the temperature and pressure are similar to conditions on Earth's surface. And Sagan and Salpeter imagined what might live there. They proposed 3 types of organisms. Sinkers, small organisms that form in the upper atmosphere and slowly fall, reproducing faster than they descend, like plankton drifting in an aerial ocean. Floaters, enormous balloon-like creatures that maintain buoyancy in the dense atmosphere, possibly kilometers across, drifting on thermal currents like living weather systems. And hunters, fast, agile organisms that prey on the floaters and sinkers. It is speculation. There is no evidence of life in Jupiter's atmosphere, but the physics works. The chemistry is plausible, and the image of organisms the size of cities drifting through alien clouds, with distributed neural networks spanning their vast bodies, processing information in ways utterly unlike anything on Earth, is one of the most haunting possibilities in all of astrobiology. Gravity is not the only planetary parameter that shapes life. Consider the rotation rate of a planet. Earth rotates once every 24 hours, giving us day and night cycles that have driven the evolution of circadian rhythms in virtually every organism. A slowly rotating planet might have days that last months. Organisms would need to cope with extended periods of intense heat and radiation, followed by extended periods of freezing darkness. Their biology would be fundamentally different from ours. Consider axial tilt. Earth's 23.5-degree tilt gives us seasons. A planet with extreme axial tilt would have extreme seasons, poles that swing from constant daylight to constant darkness, equatorial regions that become the only stable habitats, organisms migrating across continents to follow the light. Consider magnetic fields. Earth's magnetic field shields us from the solar wind, the constant stream of charged particles pouring out of the sun. Without that shield, the atmosphere would be stripped away over time, as happened to Mars. A planet without a strong magnetic field would bathe its surface in radiation. Life would need to be radiation resistant, like Deinococcus radiodurans, or would need to live underground or underwater. In October of 2025, a study from the Austrian Academy of Sciences modeled the conditions required for complex life to develop. Their analysis suggested that the specific combination of planetary parameters needed, the right gravity, the right atmosphere, the right magnetic field, the right rotation, the right distance from the right kind of star, is extraordinarily rare. They estimated that the nearest alien civilization could be 33,000 light-years away, not because life itself is rare, but because the specific conditions that allow life to become complex and technological are vanishingly uncommon. That is a sobering number. 33,000 light-years. Even at the speed of light, a message would take 33,000 years to travel that distance. The civilization that sent it might not exist by the time it arrived. But gravity's influence on alien life is just one piece of the puzzle. There is another factor that would transform alien worlds in ways you can see from space. A factor so obvious and so visual that it would be the first thing you noticed if you could look at an alien planet through a powerful enough telescope. The color of its plants. Not green. Perhaps not any color you have ever seen. That is next. Chapter 8. What color are alien plants? Why are plants green? It seems like such a simple question, but the answer reveals something profound about the relationship between life and starlight. And once you understand it, you will never look at a forest the same way. Plants are green because of chlorophyll, the molecule that captures sunlight and converts it into chemical energy through photosynthesis. Chlorophyll absorbs red light and blue light very efficiently, but it reflects green light. It bounces green wavelengths back into the world, which is why leaves look green to our eyes. Now, here is the critical detail. Chlorophyll evolved to work with the light our sun produces. Our sun is a G-type main sequence star, a yellow dwarf. It emits most of its energy in the visible spectrum, with a peak in the green-yellow range. Chlorophyll is tuned to capture the wavelengths that are most abundant in our sun's output. It is an antenna built for a specific signal. Change the star and you change the signal. Change the signal and you change the antenna. Change the antenna and you change the color of life. The most common stars in our galaxy are not yellow dwarfs like our sun. They are red dwarfs, M-type stars, smaller, cooler, dimmer, and far more numerous. Roughly 70% of all stars in the Milky Way are red dwarfs. They emit most of their energy in the red and infrared wavelengths. Visible light, the kind our eyes can see and our plants can use, is much less abundant. A plant on a world orbiting a red dwarf would face an energy crisis. There is less visible light available. The light that does arrive is shifted toward the red end of the spectrum. A plant using Earth-style chlorophyll would be poorly adapted. It would be trying to harvest light that is not there. Evolution would push in a different direction. On a red dwarf world, the optimal strategy might be to absorb every photon available. Not just red and blue, but green as well. Every wavelength. If a plant absorbs all visible light and reflects nothing, it appears black. Black plants, forests of black vegetation under a dim red sun. It sounds like science fiction, but the physics is straightforward. Scientists who model photosynthesis under different stellar spectra consistently find that plants on red dwarf worlds would likely be very dark. Black, deep purple, or dark brown, absorbing every scrap of light they can get. Now imagine the opposite, a hotter star. A blue-white A-type star that pours out intense ultraviolet and blue light. Too much energy can be as dangerous as too little. On Earth, excess ultraviolet radiation damages DNA. Plants on a world orbiting a hot, bright star would need to protect themselves. They might be highly reflective, silver or white surfaces that bounce harmful radiation away, while selectively absorbing only the wavelengths they need. Or they might be blue, absorbing the abundant blue light for energy, while reflecting longer wavelengths. The color of alien vegetation is not just a curiosity, it is a potential biosignature, one of the ways we might detect life on exoplanets from light years away. On Earth, vegetation produces a distinctive feature in reflected light called the vegetation red edge. Plants absorb visible light for photosynthesis, but strongly reflect near-infrared light. When you look at Earth's light spectrum, there is a sharp jump in reflectivity at the boundary between visible red and near-infrared. This jump is so distinctive that it can be detected from space. If an alien civilization were observing Earth from a distant star, the vegetation red edge would be one of the strongest signs that our planet hosts photosynthetic life. On an alien world, the edge would be different, shifted, occurring at different wavelengths depending on the star and the biology. But the principle would be the same, a sharp change in reflectivity caused by organisms harvesting light. Future telescopes, including the proposed Habitable Worlds Observatory, might be able to detect these features on exoplanets, seeing the color of alien forests from dozens of light years away. But photosynthesis itself might work differently on other worlds. Earth photosynthesis uses water and carbon dioxide as raw materials, powered by visible light. The process splits water molecules, releasing oxygen as a waste product. That oxygen transformed Earth's atmosphere and made complex animal life possible. But there are alternatives. Some bacteria on Earth already perform photosynthesis using hydrogen sulfide instead of water. Instead of releasing oxygen, they release sulfur. Others use iron compounds. Others use hydrogen gas. These alternative photosynthetic pathways are ancient. Some may predate the oxygen-producing photosynthesis that dominates our planet today. On a world where water is scarce but hydrogen sulfide is abundant, perhaps near volcanic vents, an entirely different form of photosynthesis could dominate. The atmosphere would contain no free oxygen. Instead, it might be rich in sulfur compounds. Animals, if they existed, would breathe sulfur-based chemistry. The entire ecosystem would run on a different fuel. Some Earth bacteria can photosynthesize using infrared light. Not the visible light that powers green plants, but longer wavelengths that our eyes cannot see. This is called anoxygenic photosynthesis. On a red dwarf world where most of the stellar output is in the infrared, this kind of photosynthesis would have a massive advantage. Organisms that can harvest infrared radiation would dominate. Plants that are invisible to human eyes, but thriving under wavelengths we cannot perceive. Consider a tidally locked planet. Many planets in the habitable zones of red dwarf stars are thought to be tidally locked, meaning one side permanently faces the star and the other side is in perpetual darkness. The day side would be blasted by constant radiation. The night side would be frozen. Between them, a ring of twilight. The terminator zone. In this narrow band, temperatures would be moderate. Light would arrive at a low angle, like a permanent sunset. Photosynthetic life might cluster exclusively in this ring. Tall organisms could lean toward the light. Motile organisms, things that blur the line between plant and animal, might slowly migrate to optimize their exposure. An entire biosphere concentrated in a thin strip around the planet, a ring of life visible from orbit. The concept of alien forests gets even stranger when you consider ecosystems that do not depend on photosynthesis at all. On a world like Europa, where the ocean is sealed beneath kilometers of ice with no sunlight reaching the water, the entire food chain would be chemosynthetic, powered by hydrothermal vents, chemical energy from the planet's interior. Such an ecosystem would look nothing like anything on Earth's surface. No plants, no photosynthesis, no oxygen. The base of the food chain would be microbial mats, feeding on hydrogen and sulfur from the vents. Whatever ate those microbes, and whatever ate those eaters, would live in total darkness. They might navigate by chemical gradients, by sound, by electric fields, by senses we have no name for. And in the thick atmospheres of gas giants or super-Earths, photosynthetic organisms might not be rooted in soil at all. They might float. Balloon, like structures drifting at the altitude, where light and pressure are optimal, rising and falling with temperature changes, reproducing by budding, dying, and sinking into the abyss below. Aerial plankton in a sky ocean. The color of alien plants is really a question about the relationship between life and its star, between biology and physics, between what evolution can do and what the environment demands. And the answer, as with so many things in astrobiology, is that the range of possibilities is far wider than our single example suggests. Green is the color of life under our sun, but under other stars, life might paint its world in colors we have never seen. Black forests under red suns, silver meadows under blue-white blazes of light, bioluminescent undergrowth glowing in the perpetual twilight of a tidally locked world. Every planet is a canvas, and evolution is the artist, working with whatever light is available, whatever chemistry is present, whatever gravity demands, painting life in colors dictated not by preference but by physics. But color is only the surface. Beneath the surface, a deeper question lurks. Would alien life follow the same patterns as life on Earth? Would evolution, given a completely different starting point, produce creatures that look anything like the ones we know? The answer might surprise you, because evolution on Earth has already run that experiment, again and again and again. Chapter 9. Evolution's repeating patterns. Look at a shark, now look at a dolphin. Now look at an ichthyosaur, a marine reptile that went extinct 66 million years ago. 3 animals separated by hundreds of millions of years of evolution. 3 completely different lineages. Sharks are fish, dolphins are mammals, ichthyosaurs were reptiles. They share no recent common ancestor, and yet they look almost identical. Streamlined bodies, pointed snouts, dorsal fins, powerful tail fins, smooth skin designed to reduce drag. The same hydrodynamic shape evolved independently 3 separate times. This phenomenon has a name: convergent evolution, and it is everywhere on Earth. The eye has evolved independently at least 40 to 50 times, not variations on a single eye design, entirely separate inventions of vision. Insects evolved compound eyes with thousands of tiny lenses. Vertebrates evolved camera eyes with a single lens focusing light onto a retina. Scallops evolved mirror eyes that use curved reflective surfaces to focus images. Box jellyfish have 24 eyes of 4 different types. Each of these visual systems was built from scratch by a different branch of the tree of life. Flight has evolved independently at least 4 times. Insects were first, roughly 350 million years ago. Pterosaurs evolved flight separately, roughly 230 million years ago. Birds evolved it again, roughly 150 million years ago. Bats evolved it a 4th time, roughly 50 million years ago. 4 completely unrelated groups of animals all arrived at the same solution to the same problem: how to move through air. Echolocation evolved independently in bats and dolphins. 2 groups of mammals that could not be more different in their lifestyles arrived at the same navigational technology: emit high-frequency sounds. Listen for the echoes. Build a mental map of the environment from the returning signals. Electric organs evolved independently in multiple unrelated groups of fish. The ability to generate electric fields for navigation, communication, and hunting arose separately in electric eels, electric rays, elephant fish, and several others. The camera eye, the wing, the streamlined body, echolocation, bioluminescence, venom, armor plating, warm-bloodedness. Evolution keeps reinventing the same solutions because physics constrains what works. There are only so many efficient shapes for moving through water, only so many ways to generate lift in air, only so many methods for detecting electromagnetic radiation. The laws of physics are the same everywhere, and evolution, regardless of its starting point, tends to converge on the same answers to the same physical problems. The biologist Simon Conway Morris has argued that this pattern suggests something profound about the nature of evolution. He believes that evolution is not random, it is channeled, constrained by physics and chemistry into a limited number of viable solutions. Given enough time and the right conditions, evolution will find these solutions repeatedly. If Conway Morris is right, the implications for alien life are remarkable. It would mean that aliens might not be as alien as we imagine. On any world with liquid water and a similar range of physical challenges, evolution might produce organisms with eyes, with streamlined bodies, with some form of locomotion through fluid. The details would differ, the underlying chemistry might differ, but the shapes, the strategies, the solutions might be hauntingly familiar. But there is a powerful counterargument, and it comes from one of the most influential evolutionary biologists of the 20th century. Stephen Jay Gould proposed a famous thought experiment. Imagine you could rewind the tape of evolution to the very beginning and let it play again. Would you get the same result? Gould said no. He argued that evolution is deeply contingent, dependent on accidents, random mutations, chance extinctions, asteroid impacts, ice ages, volcanic eruptions. Change any one of these events and the entire trajectory changes. Replay the tape and you might get a world full of intelligent cephalopods instead of primates, or a world where nothing more complex than bacteria ever evolves. The convergent evolution argument has a weakness that Gould would have been the first to point out. Every instance of convergence on Earth happened within a single tree of life. Sharks, dolphins, and ichthyosaurs all evolved streamlined bodies, but they all share the same underlying genetics, the same DNA, the same protein-building machinery, the same cellular architecture. They are variations on a theme, not independent experiments. Would convergence still occur if the underlying chemistry were different? If life were built on silicon instead of carbon? If it used ammonia instead of water? If it had an entirely different genetic system? We do not know. One data point cannot establish a universal pattern. Consider a specific question. On a world covered in liquid methane at -170 degrees Celsius, with silicon-based chemistry and an atmosphere of nitrogen, would evolution produce eyes? The answer depends on whether there is a selection pressure for detecting electromagnetic radiation in that environment. Methane is transparent to some wavelengths. If organisms could gain an advantage by sensing those wavelengths, evolution might produce something eye-like, but the structure would be radically different. The chemistry would be unrecognizable. It would be an eye in function but not in form. Or organisms might not develop vision at all. They might navigate entirely by chemical sensing, the way many deep ocean creatures on Earth do, or by vibration, or by magnetic fields. On a world without light, there is no selection pressure for eyes. Evolution would invest its energy elsewhere. The honest answer is that we cannot predict what alien evolution would produce. We can make educated guesses based on physics. We can identify solutions that seem universal because they are dictated by physical law. But we cannot know whether the specific path life takes on another world would converge with ours or diverge in ways we cannot imagine. And that brings us to the most uncomfortable question in all of evolutionary biology: intelligence. Intelligence has evolved multiple times on Earth. Primates, dolphins, elephants, crows, octopuses. Each of these lineages developed significant cognitive abilities independently. Problem-solving, tool use, social learning, self-awareness in some cases. The convergent evolution of intelligence suggests that big brains are a good solution to certain evolutionary challenges: complex environments, social competition, variable food sources. But there is a critical distinction between intelligence and technology. Intelligence has evolved multiple times. Technology has evolved exactly once. Out of the millions of species that have existed on Earth over 4 billion years, exactly one has developed technology. Exactly one has built civilizations, written languages, launched spacecraft. Us. Why? It is not enough to be smart. Dolphins are extraordinarily intelligent. They have complex social structures. They communicate in sophisticated ways, but they have no hands. They cannot manipulate objects. They cannot build tools. Their intelligence, however impressive, cannot translate into technology without manipulative appendages. Elephants have trunks that can manipulate objects with remarkable precision, but they have relatively short reproductive spans and small population densities that limit the accumulation and transmission of cultural knowledge. Crows use tools. They solve complex puzzles. But their brains are small, limited by the size of their bodies. There is a physical ceiling on how large a bird brain can grow and still allow the bird to fly. Octopuses are perhaps the most alien intelligence on Earth. Most of their neurons are in their arms, not their heads. They can solve mazes, open jars, use tools, but they are solitary. They die shortly after reproducing. They cannot pass knowledge to the next generation. Every octopus starts from scratch. The specific combination that produced technological civilization, large brains, manipulative hands, social structures, long lifespans, language, cumulative culture, required a very particular sequence of evolutionary events. It took nearly 4 billion years, and it happened once. That is a sobering fact for the search for intelligent alien life. Complex life may be common. Intelligence may evolve repeatedly, but the specific package that produces technology and civilization may be vanishingly rare. Even on worlds teeming with complex creatures with eyes and wings and streamlined bodies, the emergence of a species that builds radio telescopes might be a once-in-a-galaxy event. Or it might not. We simply do not know. And with a sample size of 1, we cannot calculate the odds. What we can say with more confidence is what alien life probably looks like in the most common scenario, not the dramatic scenario, not the blockbuster movie scenario, the statistically overwhelmingly likely scenario. And that scenario does not involve tentacles or spaceships. It involves something much smaller, much simpler, and in many ways, much more important. Chapter 10. The microbes are the answer. Here is a number that should change how you think about life in the universe. 80%. For roughly 80% of Earth's biological history, the only life on this planet was single-celled, microbes, bacteria and archaea. Tiny organisms, invisible to the naked eye, floating in oceans, coating rocks, drifting in the atmosphere. For more than 3 billion years, that was all there was. No plants, no animals, no fungi, no eyes, no brains, no wings, no teeth, just microbes. Complex multicellular life did not appear until roughly 600 million years ago. Mammals have been around for about 200 million years, primates for about 65 million, the genus Homo for about 2.5 million, technological civilization for roughly 300 years. If you picked a random moment in Earth's 4 billion year biological history, the overwhelming odds are that you would find a planet covered in microbes and nothing else. Not a planet waiting for complex life to arrive. A planet where microbial life is the complete, fully realized, spectacularly successful answer to the question of what biology can do. If this ratio applies to other living worlds, and there is no reason to assume it does not, then most alien life in the universe is microbial. Not because microbes are a stepping stone to something more interesting, because microbes are the main event. They are the default state of biology. Complex multicellular life is the anomaly. Intelligence is the extreme outlier. And technology is, as far as we know, a singular accident. That might sound disappointing. We want alien civilizations. We want someone to talk to. We want confirmation that intelligence is a common feature of the cosmos. Finding out that the universe is full of bacteria and nothing else might feel like a letdown. But it would not be a letdown. It would be the most important discovery in the history of science. Here is why. Right now, we have one example of life. 1. Everything alive on Earth shares a single common ancestor. We cannot know whether life is a cosmic inevitability or a freakish accident. If we find microbial life on Mars that arose independently from life on Earth, that question is answered. Life is not an accident. It is a natural process. It happens wherever the conditions are right. And if it happens twice in one solar system, it probably happens everywhere. The follow-up questions are equally profound. If alien microbes use DNA and similar biochemistry to Earth life, it would mean the chemistry of life is constrained. There may be only one way, or very few ways, to build a living system. The universe might be full of carbon-based, water-dependent, DNA-driven life, because that is the only combination that works. If alien microbes use different biochemistry, a different genetic code, different molecular machinery, it would mean the opposite. Life has multiple chemical pathways. The universe is not just alive. It is creatively, diversely, wildly alive in ways we never anticipated. Either answer rewrites everything we think we know about our place in the cosmos. Either answer changes biology, chemistry, philosophy, and theology in ways we can barely predict. But finding alien microbes is extraordinarily difficult. Microbes are small. They do not build radio transmitters. They do not light up cities. They leave subtle, ambiguous traces that are easy to miss and easy to misinterpret. The Cheyava Falls rock on Mars might contain evidence of ancient microbes, or it might not. The DMS signal on K2-18b might be produced by marine phytoplankton, or it might be produced by chemistry. The challenge of detecting microbial life, especially life that might use unfamiliar chemistry, has pushed scientists toward a radical new approach. Agnostic life detection, methods that do not assume you know what you are looking for. One approach is chemical disequilibrium. Living planets have atmospheres that are out of chemical balance. On Earth, oxygen and methane coexist in the atmosphere. This should not happen. Oxygen and methane react with each other. Left alone, one would consume the other within thousands of years. The fact that both persist simultaneously means something is continuously producing them. That something is life. Photosynthetic organisms produce oxygen. Methanogenic microbes produce methane. Without life, the atmosphere would settle into a boring, stable equilibrium. If we detect an exoplanet with an atmosphere containing gases that should not coexist, that disequilibrium itself could be a biosignature. We would not need to know what specific organisms produce the gases. We would just need to see the chemical impossibility of the combination and infer that something is keeping the system out of balance. Another approach is structural complexity. Living systems produce molecules and structures that random chemistry does not. The patterns are distinctive, too organized to be accidental, too specific to be produced by abiotic processes. If we can measure the complexity of a sample without knowing its exact composition, we might be able to identify biological origin from organization alone. A third approach involves isotopic ratios. Living organisms are picky about their atoms. They preferentially use lighter isotopes of elements like carbon and sulfur. When microbes metabolize carbon dioxide, they take up more carbon-12 than carbon-13. This leaves a distinctive, isotopic fingerprint. The Cheyava Falls rock on Mars shows a carbon isotope pattern that is consistent with biological processing. Isotopic analysis does not depend on recognizing specific molecules. It depends on recognizing the fingerprint of selectivity that life leaves behind. But the most exciting development in biosignature detection is happening not in chemistry labs, but in computer science labs. Machine learning. Artificial intelligence, trained on massive datasets of chemical information, is learning to distinguish biological from non-biological samples in ways that human scientists cannot. In 2023, researchers published a study in the Proceedings of the National Academy of Sciences showing that an AI system trained on mass spectrometry data could identify whether a sample had a biotic or abiotic origin with roughly 90% accuracy. The system did not look for specific molecules. It looked for patterns, subtle statistical regularities in the distribution of molecular weights and chemical structures that distinguish living chemistry from dead chemistry. Patterns too complex for a human to see, but detectable by an algorithm trained on enough data. Since then, the technology has advanced rapidly. Machine learning systems have been trained to detect what researchers call the ghosts of biological patterns in rocks billions of years old. Rocks that were previously considered too degraded to contain useful biosignature information. The AI found patterns that human analysis had missed. NASA is now building standardized databases to allow future rovers and landers to use AI biosignature detection in real time. Instead of sending raw data back to Earth and waiting months for analysis, a rover on Mars or a lander on Europa could run samples through an onboard AI and get an immediate assessment of whether the sample shows signs of biological origin. For the Europa Clipper mission and potential future Enceladus missions, this approach could be transformative. A spacecraft flying through the plumes of an ocean moon, collecting spray from an alien sea, running it through a mass spectrometer, and feeding the data to an AI trained to detect the signatures of life, not looking for specific molecules, looking for the statistical fingerprint of biology itself. We are building tools that can find life we have never seen before. Tools that do not need to know what alien biochemistry looks like. Tools that detect the pattern of life rather than its specific chemistry. This is not science fiction. This is happening now. The algorithms exist. The databases are being built. The missions are being planned. Within our lifetimes, possibly within the next decade, one of these tools might return a result that changes everything. But even if we find alien microbes, even if we confirm that the universe is teeming with single-celled life, the deeper mysteries remain. What would more complex alien life be like? Not just its body plan or its chemistry, but its experience, its perception, its mind, if it has one. How would an alien see the world? What would it feel? What senses might it have that we cannot even imagine? Those questions take us into the strangest territory of all. Chapter 11: Alien Senses and Alien Minds. Close your eyes for a moment and think about how you experience the world. Light entering your eyes, sound vibrating your eardrums, pressure on your skin, scents drifting into your nose, the taste of food on your tongue. 5 senses. That is how humans interact with reality. And for most of human history, we assumed that 5 senses was more or less the standard kit for any complex organism. We were wrong. On Earth alone, animals have evolved senses that we do not possess, and in most cases cannot even perceive without instruments. Sharks detect the tiny electrical fields generated by the muscle contractions of their prey. They can sense the heartbeat of a fish buried in sand from several feet away. This is called electroreception, and it is as natural to a shark as seeing is to you. Many birds, sea turtles, and bacteria can sense the Earth's magnetic field and use it for navigation. Magnetoreception, an entire compass built into their biology. Migratory birds flying thousands of miles use the planet's magnetic architecture as a map, sensing field lines as naturally as we sense up and down. Pit vipers see infrared radiation. Their pit organs detect the body heat of warm-blooded prey in total darkness. they are seeing a wavelength of light that is invisible to us. To a pit viper, a mouse in a dark field glows like a beacon. The mantis shrimp has 16 types of photoreceptor cells. Humans have 3. We see the world through combinations of red, green, and blue. The mantis shrimp perceives ultraviolet light, polarized light, and distinctions of color that our visual system literally cannot process. We do not know what the world looks like to a mantis shrimp because our brains lack the architecture to even imagine it. Every sense is an answer to an environmental question. Eyes evolved because electromagnetic radiation is a useful source of information. Ears evolved because pressure waves carry data about the environment. Electroreception evolved because living things generate electrical fields, and detecting them provides an advantage. On another world, the environmental questions might be different, and the senses that evolve to answer them might be unrecognizable. Consider a world with a thick, opaque atmosphere where visible light never reaches the surface. On such a world, eyes would be useless. There is nothing to see. Evolution would not invest in vision. Instead, organisms might develop extraordinarily refined sonar. Echolocation, taken to a level far beyond what bats and dolphins have achieved. Imagine creatures that perceive their entire world in sound. Not just the direction of objects, but their shape, their texture, their internal structure. A sonar sense refined over billions of years could potentially see through solid objects, detecting density differences, cavities, the internal organs of other organisms. An alien with advanced echolocation might know you are sick before you do, hearing the irregular rhythm of your heart or the density change in your bones. On a world near a magnetically active star, where magnetic fields are strong and constantly shifting, organisms might develop a magnetic sense so refined that they perceive the planet's field the way we perceive light. A magnetic landscape overlaid on the physical one. Field lines as vivid as colors. Magnetic anomalies as noticeable as shadows. On a world drenched in radiation, organisms might evolve sensitivity to X-rays or gamma rays, seeing through matter the way we see through glass. The alien equivalent of a doctor's X-ray machine might be a living sense organ, evolved naturally because the environment was saturated with high-energy radiation. On a world with complex atmospheric chemistry, chemoreception, the sense of smell and taste taken to extremes, might be the dominant way of experiencing reality. Organisms that read chemical gradients in the air the way we read light patterns. They would perceive a landscape of scent as detailed and rich as our visual world. Every breeze carrying a symphony of chemical information. These possibilities matter because they determine not just how aliens would perceive the world, but how they would communicate. And communication determines whether we could ever detect them. We search for alien civilizations by listening for radio signals. We do this because radio is how we communicate over long distances. The search for extraterrestrial intelligence, SETI, has been scanning the skies for radio emissions for over 60 years. So far, nothing confirmed. But radio has only been our dominant communication technology for about a century. Before that, we used smoke signals, drums, written messages, telegraph wires. In the future, we might move to laser-based communication, which is more efficient and harder to detect. We might use quantum encrypted channels that are by definition undetectable to eavesdroppers. If an alien civilization went through a similar technological progression, their radio phase might have lasted only a century or two. A brief window in a civilization's history when they happen to be broadcasting on frequencies we can detect. Before that window, they are invisible to us. After that window, they are using technologies we cannot detect. We would need to be listening at exactly the right time, pointed at exactly the right star. And that assumes they use electromagnetic radiation to communicate at all. A civilization of creatures that perceive the world through chemical sensing might communicate chemically. releasing complex molecules into the environment that carry encoded information. Their equivalent of language might be a biochemical signal, undetectable by any radio telescope ever built. A civilization of sonar-based organisms might communicate through structured sound patterns transmitted through their ocean or atmosphere. Their internet might be acoustic. Their broadcasts might be pressure waves that dissipate before leaving their planet. In 2023 and 2024, researchers began a project that illuminates just how alien, non-human communication can be. They call it Whale SETI. The project studies humpback whale communication systems as a model for non-human intelligence communication. Humpback whales produce complex, structured vocalizations that change over time, are culturally transmitted, and vary between populations. Their songs contain patterns that suggest syntactic structure, a form of grammar. The goal is not to claim that whales are as intelligent as humans. It is to develop the tools and frameworks needed to recognize and decode communication systems that are fundamentally different from our own. If we cannot understand the communication of another intelligent species on our own planet, a species that shares our biology, our ocean, our evolutionary history, how will we recognize the communication of a truly alien intelligence? And that leads to perhaps the deepest question. Would we recognize alien intelligence at all? Intelligence is a process, not a substance. It is not about what a mind is made of. It is about what it does. Human intelligence runs on networks of neurons connected by synapses. But octopus intelligence runs on a fundamentally different architecture. Most of an octopus's neurons are in its arms, not its head. Each arm has a degree of autonomous processing. The octopus is a distributed intelligence, with decision-making spread across its body. And it is demonstrably intelligent. It solves puzzles. It uses tools. It recognizes individual humans. It plans. Now imagine intelligence distributed not across a single body, but across a colony. A species where individual organisms are relatively simple, but the colony as a whole displays complex intelligent behavior. We see hints of this on Earth. Ant colonies and termite mounds exhibit collective behaviors that no individual ant or termite could produce. They build complex structures, manage resources, wage wars, farm fungus. The intelligence, if you can call it that, is an emergent property of the system rather than a property of any individual. On an alien world, this principle could be taken much further. A biological neural network spread across thousands or millions of interconnected organisms, collectively processing information at a level that dwarfs any individual brain. A living, thinking ecosystem. An intelligence that has no center, no skull, no single point you could point to and say, the mind is here. How would you recognize such an intelligence? How would you communicate with it? How would you even know it was there? If alien intelligence looks nothing like what we expect, if it thinks in ways that have no human analog, if it communicates through channels we cannot detect and perceives reality through senses we do not possess, first contact might have already happened. Signals in our data that we dismissed as noise. Patterns we cannot see because we are not built to see them. The gap between human intelligence and octopus intelligence is small, compared to the potential gap between human intelligence and truly alien intelligence. We are two branches on the same tree of life, separated by hundreds of millions of years of divergent evolution, but still sharing the same DNA, the same cellular machinery, the same fundamental biology. An intelligence that arose independently on another world, built on different chemistry, shaped by different physics, might be as far from us as we are from an ant. Further, the distance might be unbridgeable, and yet we keep searching. We keep listening. We keep sending probes and building telescopes and scanning the sky. Because the alternative, assuming we are alone without ever seriously checking, is not science. It is surrender. But the search raises an uncomfortable question, one that has haunted astronomers since a summer lunch in 1950. If intelligent civilizations arise in the universe with any regularity at all, the universe is old enough and large enough that we should see evidence of them everywhere. We do not. The sky is silent, and the silence has a name. Chapter 12. The Great Filter. In the summer of 1950, the physicist Enrico Fermi was having lunch with colleagues at Los Alamos National Laboratory. The conversation turned to flying saucers, which were in the news at the time. Someone made a joke. Fermi did some quick mental arithmetic, and then he asked a question that has haunted science ever since. Where is everybody? The logic behind the question is simple and devastating. The Milky Way galaxy contains roughly 200 to 400 billion stars. Many of them are older than our Sun. Many have planets. Many of those planets sit in habitable zones. If even a tiny fraction of those planets developed intelligent life, and if even a tiny fraction of those civilizations developed the ability to travel between stars or simply to send signals across space, then the galaxy should be teeming with evidence of extraterrestrial intelligence. The universe is approximately 13.8 billion years old. Our Sun is about 4.5 billion years old. Many stars in the galaxy are billions of years older. Even at a fraction of the speed of light, a civilization that arose a billion years before us could have explored or colonized the entire galaxy by now, many times over. And yet we see nothing. No signals, no probes, no megastructures, no evidence of engineering on any scale. The sky, to every instrument we have ever built, is silent. This is the Fermi paradox, and it has been gnawing at scientists for 75 years. In 1961, astronomer Frank Drake formalized the question into an equation. The Drake equation is not really an equation in the mathematical sense. It is a framework for thinking about the problem. It multiplies together a series of factors: the rate of star formation, the fraction of stars with planets, the fraction of planets that could support life, the fraction of those where life actually arises, the fraction where life becomes intelligent, the fraction that develops detectable technology, and finally, the length of time such civilizations survive. The first few factors are now reasonably well known. Stars form regularly. Most have planets. Many planets are in habitable zones. But the later factors are completely unknown. We do not know how often life arises. We do not know how often it becomes intelligent, and we have no idea how long technological civilizations last. Depending on what numbers you plug in, the Drake equation gives answers ranging from 0, we are alone, to millions of civilizations in our galaxy right now. The equation does not solve the paradox. It frames it, and the frame reveals that the answer depends on factors we cannot yet measure. One of the most influential attempts to explain the Fermi paradox is the concept of the Great Filter. The idea, proposed by economist Robin Hanson in 1998, is that somewhere between dead chemistry and a galaxy-spanning civilization, there is at least one step that is extraordinarily unlikely, a filter that almost nothing gets through. The question is where the filter sits. If the Great Filter is behind us, that is good news. It means that one of the early steps, the origin of life, the evolution of complex cells, the development of multicellular organisms, the emergence of intelligence, was the hard part. We already passed it. We are the lucky ones who made it through. The reason we do not see alien civilizations is that the step we already completed is so improbable that almost nobody else has managed it. If the Great Filter is ahead of us, that is very bad news. It means that civilizations regularly arise, reach something like our level of development, and then destroy themselves. Nuclear war, climate collapse, artificial intelligence catastrophe, engineered pandemics, resource exhaustion, some recurring catastrophe that technological civilizations cannot avoid, a trap built into the structure of technology itself. This is why the search for alien microbes is so consequential. If we find microbial life on Mars that arose independently, it means the origin of life is not the filter. Life starts easily. That pushes the filter later in the sequence, possibly ahead of us. If we find complex multicellular life on another world, the filter is pushed even later. And if we ever detect an intelligent signal from a distant civilization, it means every step up to and including intelligence is not the filter. The filter must be later still. It must be something that happens to civilizations after they develop, something that ends them. Paradoxically, finding alien life would be terrifying because it would mean the great filter is probably still in our future. In October of 2025, researchers from the Austrian Academy of Sciences published a study that modeled the probability of technological civilizations in the Milky Way. Their analysis was sobering. They estimated that the nearest alien civilization could be approximately 33,000 light-years away. For even 10 civilizations to coexist in the galaxy at the same time, the average civilization would need to survive for more than 10 million years. 10 million years. Human civilization is roughly 10,000 years old. Industrial civilization is roughly 300 years old. We have been capable of destroying ourselves with nuclear weapons for about 80 years. The gap between where we are and where we would need to be to show up in those models is staggering. There is a key insight buried in that number. If we ever detect an alien civilization, it is almost certainly much older than we are. Any civilization we find has already survived whatever filters we have not yet faced. They would be the winners, the survivors, the species that figured out how to last, and their existence would be proof that lasting is possible. But there are other solutions to the Fermi paradox beyond the great filter. Some are comforting, some are deeply unsettling. The zoo hypothesis proposes that advanced civilizations are aware of us but have chosen not to make contact. We are being observed, like animals in a nature preserve, by intelligences sophisticated enough to watch without being detected. This is unfalsifiable and therefore frustrating, but it is not illogical. If a civilization millions of years more advanced than ours wanted to observe us without interference, they could probably do it. Another possibility is that civilizations do not communicate via radio. They have moved to technologies we cannot detect. Neutrino beams, gravitational wave modulation, quantum entanglement networks. If advanced civilizations graduate from radio within a few centuries of inventing it, and if the galaxy is full of civilizations that passed that stage millions of years ago, then SETI's radio telescopes are listening on a channel that nobody uses anymore. A more unsettling possibility is that civilizations transcend. They develop artificial intelligence, upload their consciousness into digital substrates, and lose interest in the physical universe. They turn inward, creating virtual realities more compelling than outer space. Or they merge into forms of existence so different from biological life that we would not recognize them as alive, let alone intelligent. They are everywhere but invisible, like dark matter, present but undetectable by any instrument we know how to build. In 2025, researchers proposed what they called the Escatian hypothesis. The idea is that first contact, when it happens, will be a more extreme and unusual event than subsequent contacts. The first civilization we encounter will be an outlier, not a representative sample. It might be extremely old, extremely different, extremely advanced. The very fact that it is the first one we find means it is unusual in some way. Perhaps it is the only one within range. Perhaps it is the only one that chose to be detectable. And then there is the most unsettling possibility of all. The universe may be full of intelligence that simply does not care about us. We assume that advanced civilizations would be curious, that they would explore, that they would want to communicate. But those are human motivations projected onto alien minds. A civilization that has existed for 1 billion years might have concerns so far beyond our comprehension that contacting a species that just invented radio is about as interesting to them as contacting an ant colony is to us. Not hostile, not protective, just indifferent. The Fermi paradox does not have a solution. It has dozens of proposed solutions and no way to determine which is correct without more data. But the search continues, driven by the understanding that the answer matters enormously, not just intellectually, existentially. If we are alone, we carry something precious and irreplaceable. If we are not alone, we have neighbors. And the nature of those neighbors, whether they are microbes or minds, or something we cannot imagine, determines what the future holds. So what would first contact actually look like? Not in the movies, not in our imaginations. In reality, what does the science say about how we would actually encounter alien life for the first time? That is next. Chapter 13. First Contact. Forget everything the movies have taught you. First contact will not be a flying saucer landing on the White House lawn. It will not be a transmission in English saying "we come in peace." It will not be a fleet of ships darkening the sky. Those are stories. They are entertaining stories, but they have almost nothing to do with what science tells us first contact would actually look like. The most likely scenario is this. A scientist sits in front of a computer screen. They are analyzing data, spectra from an exoplanet's atmosphere, chemical results from a Mars rock sample, mass spectrometry readings from a plume sample on an ocean moon. The data shows an anomaly, a pattern that does not fit any non-biological explanation. The scientist frowns, checks the instruments, runs the analysis again, gets the same result. They send the data to colleagues. The colleagues check the instruments independently, run their own analysis, get the same result. A paper is written. It goes through peer review. Reviewers push back, ask for alternative explanations. The authors address the criticisms. The paper is published. The scientific community debates. Other teams try to replicate the finding. Some succeed. Some get different results. Years pass. Gradually, the evidence accumulates. The alternative explanations are tested and eliminated one by one. And at some point, slowly, without fanfare, the consensus shifts. We are not alone. That is how it would happen. Not with a bang, with a data point. We are already living through the early stages of this process. The Cheyava Falls rock on Mars was discovered in July of 2024. The peer-reviewed paper was published in September of 2025. Over a year of analysis between discovery and publication. And the debate is far from over. Scientists are still discussing alternative explanations. More data is needed. The samples need to be brought back to Earth for definitive analysis. That requires a Mars sample return mission that has not yet launched. The K2-18b dimethyl sulfide detection followed the same trajectory. A tentative hint in 2023, a stronger signal in 2025, a NASA-led reanalysis that found no conclusive evidence. Independent teams getting different results depending on their methods and models. The 5 sigma confirmation threshold still unmet. More observation time needed. This is what the search for life actually looks like. Not a single dramatic moment. A long, careful, frustrating accumulation of evidence. And that is exactly how it should be. The stakes are too high for anything less. But the type of life we find first would dramatically shape the consequences. Microbial life on Mars or Europa would be revolutionary for biology, but manageable for society. It would confirm that life is common in the universe. It would answer one of humanity's oldest questions. But it would not threaten anyone. Microbes do not invade. They do not communicate. They do not pose military challenges. The discovery would be celebrated, debated, studied, and eventually absorbed into the scientific mainstream. Textbooks would be rewritten. Funding for astrobiology would increase. Life would go on. But microbial life on Mars would make the Fermi paradox sharper. If life starts easily, why do we not see anyone? Where is everybody? The great filter becomes more ominous. An intelligent signal from a distant star would be a completely different category of discovery. Every religion on Earth would need to grapple with the implications. Every philosophy would need revision. Every assumption about humanity's special place in the cosmos would be challenged. And there is the practical problem of communication. If the signal comes from a civilization 33,000 light-years away, as the Austrian Academy study suggests is likely, a message would take 33,000 years to reach them. Their reply would take another 33,000 years to return. A single exchange would span 66,000 years, longer than all of recorded human history. A conversation would be impossible. We could receive their signal and know we are not alone, but we could never have a dialogue. Stephen Hawking warned that contacting an alien civilization could be dangerous. He drew an analogy to the European colonization of the Americas. When a technologically advanced civilization encounters a less advanced one, the result is rarely good for the less advanced party. Others have countered that our radio signals have been leaking into space for over a century already. If anyone is listening within 100 light-years, they already know we are here. The horse has left the barn. But the most realistic first contact scenario may not involve communication at all. If the universe is as old and as large as we know it to be, and if other civilizations exist, the vast majority of them are either much older or much younger than us. Given the age of the universe, much older is far more likely. A civilization that has survived for millions of years would have technology so advanced that we could not distinguish it from magic. And if they sent probes to explore the galaxy, those probes might have arrived in our solar system long ago. We might encounter not a living civilization, but an artifact, a machine, a probe, something built and sent by a species that may no longer exist. Even a silent artifact would reveal volumes about its creators. Its materials would tell us about their chemistry. Its engineering would tell us about their physics. Its design choices would tell us about their priorities. We would learn from it even if we could never speak to whoever built it. The Breakthrough Listen initiative, the most comprehensive SETI search ever conducted, is monitoring millions of stars across multiple wavelength bands. So far, nothing confirmed. But the search has only covered a tiny fraction of the sky, and the sensitivity of the instruments continues to improve. Absence of evidence is not evidence of absence, especially when you have only checked a small part of the haystack. Meanwhile, the search for microbial life is accelerating on multiple fronts. Europa Clipper is en route to Jupiter. The European Space Agency's Rosalind Franklin rover, designed to drill beneath the Martian surface where organic molecules might be shielded from radiation, is expected to launch in 2028. ESA is planning a dedicated mission to Enceladus that could fly through the plumes and sample the ocean directly. The TRAPPIST-1 system, a red dwarf star about 40 light-years away, has 7 rocky planets. 3 of them orbit in the habitable zone. JWST is analyzing their atmospheres right now. In October of 2025, researchers announced the discovery of a super-Earth, less than 20 light-years away, that could be another candidate for atmospheric characterization. Every new telescope, every new mission, every new analysis technique narrows the search space and increases the probability that if life is out there, we will find it. But there is a possibility we rarely discuss. What if we find something and cannot understand it? If the alien intelligence is more advanced than us, which the age of the universe makes overwhelmingly likely, it may have transcended biology entirely. We might meet machines, artificial intelligences sent as probes, self-replicating systems that have been exploring the galaxy for millions of years without any biological crew. Their intelligence might be incomprehensible to us. Not because they are withholding information, but because the gap between our minds and theirs is too large to bridge. As large as the gap between us and an insect. We might find something and know that it is artificial, know that it was made by intelligence, and still have no idea what it means. A message we cannot decode, a signal we cannot interpret, a structure we cannot understand. First contact might be the discovery not of an answer, but of a deeper question, proof that intelligence exists in the universe beyond our own, paired with the humbling realization that we may never understand it. That is a possibility we need to prepare for, not just scientifically but psychologically. The search for alien life is ultimately a search for meaning, and the meaning we find might not be the meaning we expected. Which brings us to the final question, the one that underlies everything we have discussed tonight. The one that matters regardless of whether we find microbes on Mars, molecules on K2-18b, oceans on Europa, or silence stretching from here to the edge of the observable universe. What does the answer mean? Chapter 14. What the answer means. We have come a long way tonight. From the leopard spots on a Martian rock to the dimethyl sulfide drifting in the atmosphere of a world 124 light-years away. From the hydrothermal vents at the bottom of Earth's oceans to the geysers erupting from a tiny moon of Saturn. From the chemistry of silicon and methane to the crushing gravity of super-Earths. From black forests under red suns to sonar-based minds perceiving reality in ways we cannot imagine. From the great filter to the silence of the sky. And now we have to sit with the question that has been waiting for us all along. What if we find life? What does it mean? And what if we do not? Let us take the first possibility. Suppose we find microbial life on Mars. Simple organisms, single-celled, perhaps already dead for billions of years, preserved as chemical ghosts in ancient mudstone. Perhaps alive, clinging to existence deep underground where liquid water still seeps through Martian rock. This discovery would tell us that the origin of life is not a miracle, it is a natural process, something that happens when the right chemistry meets the right conditions. If life arose independently on two planets in the same solar system, the implication is inescapable. Life is common. The universe is alive, not in every corner, not on every rock, but frequently enough that a cosmos without life would be the anomaly, not a cosmos with it. That changes everything, not just biology, philosophy, theology, our understanding of our own significance. For 400 years, the Copernican revolution has been slowly dismantling the idea that we are special. The Earth is not the center of the solar system. The Sun is not the center of the galaxy. The galaxy is not the center of the universe. Finding that life arose independently elsewhere would be the final step. We are not the point. Life is the point, and the universe makes it readily. Now suppose we find complex life, not just microbes, multicellular organisms with differentiated tissues and specialized functions, creatures with body plans, ecosystems with food webs, on Europa perhaps, in the warm dark ocean beneath the ice. This would tell us that evolution is not just a terrestrial phenomenon, it is a universal engine. Given life and time, complexity emerges. Organization builds on organization. Simple becomes sophisticated. The process that turned single cells into blue whales on Earth operates everywhere. It is written into the laws of physics, not a local accident, a cosmic principle. And if we find intelligence, a signal, a structure, an artifact that could only have been produced by a thinking mind, then consciousness is not a fluke. Awareness is not an accident that happened once on one small world. It is a natural product of sufficiently complex evolution. The universe does not just make life, it makes minds. Each level of discovery exponentially increases the significance. Microbes mean life is common. Complex life means evolution is universal. Intelligence means consciousness is written into the fabric of reality. Each answer opens a door to a larger room. But there is another possibility, and it is the one we rarely talk about, because it is the loneliest thought a species can have. What if we search and search and search and find nothing? What if the Mars samples come back and the leopard spots turn out to be purely geological? What if the dimethyl sulfide on K2-18b turns out to be produced by chemistry, not biology? What if Europa Clipper arrives at Jupiter and finds a sterile ocean? What if we fly through the plumes of Enceladus and the mass spectrometer shows nothing but water and minerals? What if SETI listens for another century and hears only static? What if we are alone? That would be the loneliest discovery in the history of discovery, the confirmation that in all the billions of galaxies, across all the billions of years, on all the billions of worlds, consciousness sparked exactly once, here, on this small blue rock, circling an ordinary star in the suburbs of an ordinary galaxy. But loneliness is not the right word. If we are alone, then what we have is not just rare, it is precious beyond any measure. Every thought, every conversation, every act of kindness, every piece of music, every scientific discovery, every moment of wonder is unique in the cosmos. There is no backup. There is no other example. Everything we are and everything we do exists in one place and nowhere else. And that transforms the stakes of our survival from important to sacred. If we are the only intelligence in the universe, then protecting this civilization, this species, this fragile experiment in consciousness, becomes the most important task that has ever existed or will ever exist. Not just for us, for the universe itself. Because without us, or something like us, the universe has no one to see it, no one to understand it, no one to wonder at it. It is a vast, beautiful, intricate machine running in an empty room. The responsibility of being alone would be staggering. But we do not know which scenario is true, and that uncertainty is itself a kind of gift, because it means the search still matters. The question is still open. Every mission, every telescope, every experiment is a chance to change the answer. And the missions are coming. Right now, as you listen to this, Europa Clipper is hurtling through the solar system at tens of thousands of miles per hour, carrying a radar that has already proven it can see through ice. In a few years, it will arrive at Jupiter and begin the most detailed study of an ocean world ever attempted. 49 close flybys, 9 instruments, every pass bringing us closer to knowing whether Europa's ocean could support life. The James Webb Space Telescope continues to analyze the atmospheres of exoplanets, searching for the chemical fingerprints of biology. TRAPPIST-1, K2-18b. New targets discovered every year. Each observation pushes the boundaries of what we can detect from 1 million miles away. NASA's Dragonfly drone will fly across the surface of Titan in the mid-2030s, exploring a world where methane plays the role that water plays on Earth, searching for a completely independent experiment in chemistry, a second origin of life built on rules we have never encountered. The European Space Agency's Rosalind Franklin rover will drill beneath the surface of Mars, accessing material that has been shielded from radiation for billions of years. Material that might preserve organic molecules or biosignatures that surface rocks cannot retain. AI systems are learning to detect life without knowing what it looks like, identifying the statistical fingerprint of biology in mass spectrometry data with accuracy that improves with every dataset, seeing patterns in ancient rocks that human scientists missed. The Habitable Worlds Observatory, a next-generation space telescope, is being designed to directly image Earth like planets around nearby stars, to see their light, to analyze their atmospheres, to look for vegetation, red edges, and chemical disequilibria, and all the subtle signs that a world is not just habitable but inhabited. We are not just asking the question anymore, we are building the tools to answer it. And that, in the end, may be the most extraordinary thing about our species. Not that we are intelligent, not that we build technology, but that we ask questions we do not have to ask. Questions that do not help us find food or avoid predators or raise our children. Questions that serve no survival purpose at all. Questions like, are we alone in the universe? No other species on Earth has ever asked that question. No other species has built telescopes to search for the answer. No other species has sent robots to other planets to look for the chemical traces of life. Whatever else we are, whatever our flaws, whatever our failings, we are the species that looks up and wonders. And whatever form alien life takes, if it exists, that wondering connects us to something larger than our daily concerns. It connects us to the universe itself, to the process that makes stars and planets and chemistry and biology, to the same process that made us. We might not be the only life in the cosmos. We might not be the only intelligence. We might not be the only civilization. But right now, in this moment, we are the only ones we know about. And that makes every question we ask, every signal we listen for, every rock we examine, every molecule we detect a kind of prayer, not to any deity, to the universe. Show us what you have made. Show us what else is out there. Show us that we are not the only ones wondering. The search continues, and whatever we find, the act of searching is itself a statement about what we are. We are the species that could not stop looking up, that could not stop asking, that saw the stars and felt something that no survival instinct can explain, something that drove us to build telescopes and launch probes and write equations and dream about worlds we will never visit. Whatever alien life looks like, if it exists, it will be the most important discovery in human history. And if it does not exist, then we carry something in us that is more precious than we ever knew. Either way, the night sky will never look the same again. If you enjoyed this deep journey into the science of alien life, please like this video and subscribe to the channel. Your support makes these detailed investigations possible. Tell me in the comments, what do you think alien life really looks like? And where do you think we will find it first? Mars, Europa, Enceladus, or somewhere we have not even thought of yet? These are not just questions for scientists. They are questions for all of us, because the answer, when it comes, will belong to everyone. Thank you for listening. I will see you in the next investigation.